Okay. Now, instead of coming in with a lit cigarette or something like that into this hydrogen and oxygen filled room, I could come in with a little bit of fine powder or certain types of metals. Nickel would do it, platinum would do it if you got a lot of money, palladium would do it if you got a lot of money. And if I were to do that, everything would go boom the minute I walked in. And once you picked up the pieces and the debris, if I brought in, say, a gram of powdered platinum, what, I wish. If I brought in a gram of powdered platinum and you picked up the mess after the explosion and got every little bit of platinum, you'd find I still had a gram of platinum left. Maybe scattered all over the city of Jacksonville, but would still be there. It had not gotten used up in that reaction. And that's what we see here. Those are what we call catalysts. Catalysts are molecules by a wide range of mechanisms. They're molecules that will speed up reaction rates. In some cases, 1 trillion times or even more. They'll speed up rates so much that reactions that ordinarily would not occur at room temperature, such as the hydrogen and oxygen reaction, now will occur at room temperature. So, now how and why do these things speed up reaction rates? Well, what catalysts do is they make an alternate, a different reaction pathway. The catalysts are involved in making that pathway by many different mechanisms. But that new pathway has a much lower activation energy barrier than the old pathway. So, lower activation energy, faster reaction rates. Or reactions occur at temperatures they normally wouldn't, like hydrogen and oxygen burning. It can happen at room temperature with the right catalyst. So what they do here is you have a new reaction pathway with lower activation energy. Okay, so they speed up reaction rates because they provide an alternate reaction pathway of lower activation energy, allowing reactions that ordinarily wouldn't occur at room temperature, for instance, to occur at room temperature, or allowing reactions to occur much, much, much faster than they ordinarily would. And catalysts, even though catalysts are going to participate in these reactions, they will not get used up. They may get restored at the end of the reaction, regenerated in one way or another. They don't get used up in the reactions they participate in. So catalysts can be used over and over and over and over again. All right, they do not get used up in these reactions. Okay, now a little analogy here. I guess I can get rid of this diagram. We're going to make a slightly different diagram. Okay, let's suppose we've got two cities here. We have reactant city right here. And down in the valley below is product town. Okay. Now, The problem is you want to have a bit of trade between these two cities. But the problem is, in between there is a big mountain range. Okay, in the old days, what you had to do is you had to take this trail up this whole mountain all the way up to the top nearly, and then all the way back down again. Took a lot of time, used a lot of effort. Wasn't much trade going on because they had to go over that big mountain barrier. And then they got their congressman. They decided to get their congressman and give them a lot of campaign finance money and stuff. And he said, hey, we're going to have a special project here. We're going to build a tunnel through that mountain. Put a highway in it, right? $billion of federal money. So he gets it, right? Because otherwise they won't finance his next campaign. So now we build this tunnel through the mountain. Okay, it's a lot easier to go down through the tunnel or back up through the tunnel. Takes less effort, less energy than climbing up over the mountain. So now all of a sudden trade between the two cities increases dramatically. That's an analogy for how catalysts work. An alternate reaction pathway, effectively almost like a tunnel through a mountain. So, catalysts are very powerful, very useful tools. Okay, now why do we talk about catalysts here? Well, we see catalysts used in all kinds of different processes. When I make something called a polyacrylamide gel, I'll make these with you guys work with them later on in the lab. A common way of separating nucleic acids and or proteins by size. You start out with these liquids and you have to get them to join together to form sort of a semi-solid jelly-like mass. In order for that process to happen, I have to add catalysts in that speed that process up, because otherwise that stuff would stay liquid for days, even weeks, and nothing would happen. But even though it is an exergonic reaction, but once again the activation energy of that reaction is high enough that it occurs at vanishingly low rates. We use catalysts all kinds of other things. They use catalysts to refine crude oil. And different kinds of catalysts will promote the production of motor oil, diesel oil, gasoline, whatever kind of thing. The chemical industry, to make all kinds of plastics and other things, they use catalysts up the wazoo. They use all kinds of catalysts. So we're seeing catalysts of different kinds in all kinds of industrial processes. And each catalyst works with a certain range of reactions. Some catalysts, like those powdered metals, catalyze a wide range of reactions, none of them all that well. Others are much more specific. So there's all kinds of different catalysts. Anybody who's a chemistry major has probably run into that. Anybody who goes into chemistry as a career is going to run into tons of catalysts. An awful lot of things about using catalysts to make various products. Okay, well that's fine. We see that a lot in our industrial processes. Now another thing, the idea that catalysts don't get used up in the reactions they catalyze. That's pretty useful, because if they did, it gets pretty expensive. Think of this. At the front end of your tailpipe, you've got that little box, the catalytic converter. What does that do? It breaks down nitrogen oxides, which are toxic and pollutants and stuff, and breaks it down into nitrogen and oxygen and goes out the tailpipe. Now, that catalytic converter is filled up with metals like rhodium, platinum, palladium. In other words, very expensive metals. If you have to replace one, if it breaks or falls off or whatever, it's going to set you back a good $1,000. And they even recycle the metals in those. But ordinarily, you're not going to have any problems. The catalytic converter is likely to be one of the last things to fail on your car. You can drive these things, my wife and I have driven cars for 250,000 or more miles, and of all the things we had to replace, the catalytic converters were not one of them, which is a good thing, because that's a very expensive job. So, that's telling you, it would be prohibitively expensive if you had to replace your converter every time you took a short hop to the local supermarket. Not too many people, whose name's not Gates or Buffett, would be driving around. But catalysts don't get used up in the reactions that they catalyze. So those metals and stuff in that catalytic converter, in fact, they get recycled when people replace converters. Companies take it, collect the metals, boom, into new ones. They still charge you $1,000 for it. Okay, well, so catalysts don't get used up in the reactions they catalyze. It can be done over and over and over and over and over again. Now, why do we talk so much about catalysts here? Reason why. Not only are many, if not most, not only are most reactions that occur inside cells endergonic, even a lot of the exergonic reactions won't occur at body temperatures at an appreciable rate. In order for them to occur, you need catalysts. Catalysts are not just for industrial processes. You could say, like the chemical companies, better living through chemistry, whatever. Okay, you could say, all right, not just better living through chemistry, living itself through chemistry, through use of catalysts. Because our cells and our bodies are filled up with all kinds of different catalysts. They're very specialized biological catalysts. And we give these guys a name. We call them enzymes. An enzyme is nothing more than a special catalyst that catalyzes certain chemical reactions. And they allow those reactions to occur at temperatures they otherwise wouldn't. Now, most enzymes are protein molecules, as we'll see. But there are some enzymes that are actually RNA molecules. More on that when we talk about nucleic acids. So certain RNA molecules, a limited number, actually also function as catalysts, and very important catalysts indeed, but we'll see that later. But the majority of enzymes we deal with are protein molecules. Now, like all catalysts, they participate in chemical reactions, but they do get regenerated when those reactions are over. They do it by a wide range of different kinds of mechanisms. Usually bind the molecules involved closely together and then often transfer chemical groups or sometimes electrons from one to the other to catalyze that reaction. Okay, now the thing with enzymes, it's different than our typical industrial type of catalysts, which are metals, certain kinds of ceramics, certain kinds of organic molecules. But the big difference with enzymes, our enzymes are extremely specific. Many of our commercial catalysts, especially those powdered metals like nickel and platinum and palladium, those kind of things, they catalyze a very broad range of chemical reactions, but they're not really good at any one of them. They're generalists rather than specialists. But these enzymes are very specialized. They catalyze certain reactions, sometimes just one specific reaction, and they catalyze it extremely well. Others catalyze a limited range of reactions involving similar molecules. So they're highly specialized. Each enzyme, that's why we have to have thousands of different enzymes in our cells, because each one catalyzes only a very limited range of reactions. So we'll say that enzymes are highly specific. And because they're specialists, they can be optimized to catalyze a given reaction incredibly well. So we could say highly effective or efficient. There are enzymes out there that can speed up reaction rates by 10 to the 17th power. That is 100,000 trillion fold. That's an enormous number. That is far more than the total number of seconds from the beginnings of the universe to the present day. That gives you an idea. Okay, some of these things can catalyze over 1 million reactions a second. Most don't work that fast, but there are some catalysts that can catalyze 1 million reactions a second or more in some cases. So they're incredibly good at what they do. They're specialists, but they're very good at catalyzing certain reactions. And leads us to a little bit of enzyme terminology here. And I'll give an example using a real enzyme that many of us are familiar with. Okay, enzymes are very large molecules, whether they're proteins or RNAs, and only a small region of that molecule is actually involved in catalyzing reaction. The rest of the molecule is to hold that region together in the right orientation, right shape, and for regulatory purpose. So we can turn an enzyme on, off, or ramp it up part way, ramp it down, whatever the case is. So you have an enzyme, and I'm going to draw an enzyme here. And there's a region in the enzyme where the catalytic reaction actually occurs, and that's what we call the active site of an enzyme. Now I draw it here as a pocket inside the enzyme because many times that is exactly what the active site is, a physical pocket or indentation in the enzyme molecule. Now molecules that bind to the enzyme and are going to be catalyzed by the enzyme will catalyze some kind of reaction. We call those molecules substrates. I'll put two over here, S1, S2. Okay, now the enzyme's active site happens to be shaped and have just the right kinds of chemical groups there to bind the substrates in a precise orientation and a precise location. Now, once that's done, the enzyme will catalyze a reaction. Let's suppose it's simply joining these guys together, forming a covalent bond. That's actually fairly common. So now we have a product of that enzyme. Okay, so that's a little bit of terminology. And the enzyme itself, the active site, may do any of a number of things. Chemical groups may break covalent bonds in part of the molecule. They may transfer electrons from one molecule to the next. They may tear a chemical group off of one of the substrates and transfer it to another and form a covalent bond there. Any of a number of different kinds of reactions, enzymes, different mechanisms that enzymes can use to catalyze certain reactions. Just depends on the reaction. Now, as an example here of a real-life enzyme to kind of reinforce this terminology, let's look at this enzyme here. It's called ADH, or alcohol dehydrogenase. Okay, ADH is found in all eukaryotic organisms. In most multicellular organisms, liver cells are loaded up with ADH. Now what this thing does is it takes a molecule and its substrates, in other words what it reacts with, are a limited number of small molecules with a single OH or hydroxyl group that we call alcohols. And what it does here is we have, I'll say, with a name like dehydrogenase, what it means is it tears a hydrogen off, and then you have a double covalent bond to the oxygen. So we have, and that forms a molecule, an organic molecule called an aldehyde. Okay. Now, many of us have experienced the effects of ADH. Because if you consume quantities of ethyl alcohol or ethanol, ADH is on the pathway to breaking it down. And then, so what happens here with ethanol, okay, so here's ethanol. ADH will convert it to this. And that's called acetaldehyde. And then a second enzyme turns acetaldehyde into acetic acid or vinegar. That's why sometimes in somebody overindulges, they have the sour taste in the mouth and sour breath and stuff. That's because of the acetic acid. That's a final breakdown product of ethyl alcohol. Now there is a little problem when you do that, especially if you do that in large quantities. Acetaldehyde's toxic. So you wake up the next morning and you're wondering who set the M80s off behind your eyeballs the night before. You got a headache, you feel sick to the stomach, dry mouth, the whole 9 yards. You feel miserable. You got yourself a hangover. You can thank acetaldehyde in part for giving you those nasty symptoms. And then eventually it's broken down to acetic acid. Now that's what so the substrate here of ADH is ethyl alcohol. The product is this acetaldehyde. Now the thing is, ADH has several substrates. They're all small alcohols. So for instance, if we were to take this molecule instead with a single carbon, that's what we call methanol, wood alcohol. And then what we end up with is formaldehyde. Okay, talking about getting pickled. All right, now that's one reason. That is why methyl alcohol, wood alcohol, is so toxic. Now the thing that you run in commonly that's full of methanol is your windshield wiper fluid. If you ever buy that, you look at the label, it's got this big leering skull and crossbones on it. It says, danger, poison, may be fatal or cause blindness if swallowed. Cannot be made non-poisonous. That says right in the label. Reason why, it's not so much that the methanol itself is toxic. It is in large amounts. It's what ADH breaks it down to. It breaks it down to formaldehyde, embalming fluid. And that quickly starts destroying all kinds of things. First thing that goes is the optic nerve. That's why you go blind. As little as a shot glass of methanol will blind you for life. And once in a while that happens in some countries like India and things like that, where people are selling, basically moonshine, and to be cheap and nasty about it, they'll throw in cheap methanol into the stuff. And sometimes you hear in India about, hundreds of people going blind and scores dying from that. I guess during Prohibition there was some of that in the United States too. And God only knows, I'm sure they probably do that. Probably happens in China too, as well. So that's the thing. It's not so much that the methanol itself is toxic. It's the formaldehyde produced as the product of ADH that's toxic. And matter of fact, what they do for methanol poisoning is they pump you full of as much ethanol as you can stand without actually outright dying, and they keep on pumping it into your bloodstream. And the idea is that the extra amount of ethyl alcohol ties up the ADH, so it works on the ethanol, and then there's other backup breakdown pathways that can take out the methanol and stuff. So that's what they use clinically is for methanol poisoning and stuff. It works to a limited extent. And likewise, if I took isopropanol, a 3-carbon alcohol, what we call rubbing alcohol, you see the label on that, it says will cause serious gastric disturbances if taken internally. In other words, you drink it, you're going to get very, very sick. And here again, that forms another aldehyde that is also quite toxic, and it makes you very, very sick. So, limited number, the number of substrates, a handful of small alcohols are substrates for the enzyme ADH. Very large molecules that have an OH group at the end of it, it won't work. So in this case, ADH, as an example, has a limited number of substrates. They all have the common feature of being small and having a single OH group, in other words, being an alcohol at the end of it. There are other enzymes that are so specific they will react with one substrate and nothing else. Even very closely related molecules are not sufficient. So, that's a little terminology about enzymes.